Hello, everyone, and welcome to today's webinar, Immunological Profiling Using Multiple Model Systems to Understand SARS-CoV-2 Infection. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. Now, we encourage you to participate today by submitting as many questions as you have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speakers. The first speaker, Dr. Premnath Rajakannu, will be introducing the audience to QuantiGene Plex technology and technical validation data provided by the collaborator. And our second speaker, Dr. Claudio Fenizia, will be presenting work done in Dr. Mara Biasin's lab at the University of Milan, Italy. Welcome to both of you. You may now begin your presentation.
Thank you for the kind introduction. Today we are going to talk about immunological profiling using multiple model systems to understand SARS-CoV-2 infection. Coronavirus are a large family of viruses that are known to cause various diseases in humans, camels, bats, and cattle. There are 7 different coronaviruses that are known to infect humans from the first time they were identified in the 1960s. MERS, SARS-CoV, and the 2019 novel coronavirus, also known as the SARS-CoV-2, are known to have crossed over from animals to humans. SARS-CoV-2, which is the focus of this talk, is a positive single-stranded RNA virus with a genome size of 30 kilobase pairs. The virus affects primarily the lung epithelial cells of the host organism. The host system's heightened inflammatory response invokes a cytokine storm, causing collateral damage to the lungs and incubant pneumonia. An urgent need to study not only the viral pathogenesis, but also the adverse host immune response calls for the use of multiparametric molecular assays that can simultaneously quantify the viral and relevant host molecular markers. Luminex XMAP technology is best built on superparamagnetic particle-coated spherical microbeads. These beads carry carboxyl groups that can covalently bind with amide groups from antibodies, peptides, and nucleic acids. These beads are then filled with a specific proportion of red and far red dyes, giving rise to 100 different combinations of bead sets with a unique red to far red dye ratio. Each bead set, owing to its surface carboxyl chemistry, are coupled with target-specific capture probes and can be mixed together in a single reaction to perform multiplexing RNA or protein quantification. The beads are measured using different Luminex instruments. One type of instrument functions like a flow cytometer, where a red laser measures the red to far red dye ratio, identifying the bead set or the target, and the green laser measures the number of fluorophores bound to the beads, hence giving us the quantification of the bound target molecule. The second type of instrument is a fluorescent imager, where the lasers are replaced by LED lamps and fluorescence is measured by a CCD camera. Let me introduce you to QuantiGene Plex assay technology. QuantiGene Plex is a multiplexing RNA quantification method that combines branched DNA technology and the Luminex XMAP technology. QuantiGene is an in situ hybridization-based method where samples such as cells, tissues, or whole blood from a range of species can be directly used to quantify up to 80 RNA targets simultaneously. The samples are homogenized using specific buffers containing RNA stabilizing agents, and the target RNA is captured and quantified using multiple small DNA probes. The most prominent feature of the technology is that it does not require RNA isolation and no reverse transcription of mRNA to cDNA, like in a Qpcr method, making the method free from any enzymatic conversion bias. Here is an animation explaining how the technology works. First, a spectrally coded Luminex XMAP beads are customized to capture several mRNA targets within your desired panel. Small DNA probes specifically designed to hybridize with different regions across the mRNA sequence is allowed to cooperatively hybridize with the target mRNA molecule. Once the capture hybridization is complete, the branch DNA probes are sequentially added to generate the signal amplification step. The endpoint fluorescent signal is generated by thousands of fluorophores that bind to these branch DNA structures. By exploiting the power of XMAP technology and the highly specific branch DNA technology, we are able to reliably quantify up to 80 mRNA targets, including normalizing genes in each reaction well. To help scientists working on SARS-CoV-2 research, we now have a pre-configured QuantiGenePlex 50-plex catalog panel. Typically, QuantiGenePlex panels are custom designed and manufactured, but the significance of cytokine storm in COVID-19 disease drove us to build this panel that will help scientists investigate the host immune response while simultaneously quantifying the viral load in the host system. We have further extended the viral targets to also cover influenza and RSV viral species to help specifically understand COVID-19 related immune biomarkers in relation to other viral respiratory diseases. The panel also includes 5 carefully selected housekeeper genes that can be used for normalizing or relative gene expression signals. If the user wishes to customize this preset panel, this is certainly possible by simply adding new genes of interest to this existing list with the help of our design team. Here is the genomic map of the SARS-CoV-2 virus, Wuhan isolate. Its genome is 30 kilobase pairs long, consisting of 10 coding regions. In the QuantiGene virus host 50-plex panel, we have designed probes targeting various coding regions spanning the entire genomic sequence of the virus. Such a design gives the possibility not only to quantify the virus with high sensitivity, but also help characterize specific structural and functional genes that might be involved in the pathogenesis. 2 separate virus host panels have been designed and developed to be used in human and mouse species. As several research groups are employing both mouse and human models to understand the pathogenesis and related host immune modulations, these 2 model-specific panels can be useful in translational research. The information about the targets and ordering of these panels can be found under these links on the Thermo Fisher Scientific website. Also, more detailed information and related literature on QuantiGenePlex assays can also be found under the link you see on the slide. We collaborated extensively with research groups from the University of Milan in Italy and from the University of Tennessee, Memphis in the United States. In the next few minutes, I would like to present some validation data from our collaborators' work. On this slide, I'd like to show you the sensitivity and dilution recovery of the various SARS-CoV-2 QuantiGene probe sets. On the left is a dilution series of SARS-CoV-2 RNA from an Italian isolate, measured using the human panel. A great linearity was observed for viral target probes at very high dilutions of the viral RNA. Viral copies as little as 150 to 200 copies were measured reliably using the pan probe. On the right, you see a similar dilution series of isolated total RNA from mouse lung tissue, measured also using a mouse panel. The mouse was infected with SARS-CoV-2 virus. A signal saturation for the pan SARS-CoV-2 probe was observed at amounts less than 100 nanograms of RNA. This is reflected in the linearity coefficient values that you see on the graph, inferring that the virus can be quantified using a very little amount of total tissue RNA. Both the human and mouse panels use identical designs for the viral targets. This slide shows the signal titration of RSV probes from a sample spiked with reference RSV RNA. The measurement was performed using the human 50-plex panel. As you can see in the raw data chart, signals were measured with very good linearity exclusively for the RSV probes, while the influenza and SARS-CoV-2 probes showed mere background signals. This showcases the exemplary specificity of the probe design in the QuantiGene assay. In another experiment, the collaborators from Milan used A549 lung epithelial carcinoma cells as in vitro SARS-CoV-2 lung infection model. A549 cells were transfected with different ORFs of either human or bat coronavirus. Cells were lysed and measured using QuantiGene-plex 50-plex human panel. You can see that the transfected SARS-CoV-2 ORFs were specifically measured in the respective samples. However, ORF8 is highly conserved across bat and human coronavirus, and this can be seen by strong and comparable signals between the ORF8 human and ORF8 bat samples. As ORF3C and 7B are not a part of the panel, they do not show any signal in the transfected cells. This data from the Memphis group shows the specificity and sensitivity of the QuantiGene assay in human cell culture infection model. Primary tracheal epithelial cells were infected with different isolates of SARS-CoV-2 for 24 hours. The cells were lysed using sample-specific buffer, and the lysates were diluted and directly subjected to QuantiGene analysis. The graph on the top shows normalized gene expression of viral targets, and the bottom graph shows log-transformed normalized gene expression of host immune response targets. All three different isolates were able to infect the tracheal epithelial cells and invoke a response that was measured in a multiplexing manner from the cell lysates. This figure illustrates once again the sensitivity of the SARS-CoV-2 probes in the assay. The culture wells were used directly to prepare the lysates, and approximately 70,000 cells per well were used. The x-axis in the figure shows sample lysate dilutions, and the y-axis shows the background subtracted fluorescent signal values. As you can see, even at 1 to 200 sample dilution, the SARS-CoV-2 targets and the most host human targets were reliably quantified. Finally, this data from Milan group shows immune response to SARS-CoV-2 and influenza infection on ex vivo human PBMC infection model. Healthy human donor PBMCs were stimulated with influenza or SARS-CoV-2 virus, and the PBMCs were lysed and directly used to quantify viral and human inflammatory markers. Log fold change of normalized gene expression of flu-stimulated PBMCs over unstimulated PBMCs and SARS-CoV-2 stimulated PBMCs over unstimulated PBMCs is represented in this graph. This experiment used only 100,000 PBMCs to quantify 50 targets simultaneously in each sample. It is worthy to note that the influenza invoked a stronger inflammatory response compared to the SARS-CoV-2 infection. This now nicely sets the stage for me to hand over the talk to our collaborator, Dr. Claudio Fenizia from the University of Milan.
Start. Thank you for the introduction. I will now take over and present you some of our results. Upon virus detection, our body quickly reacts and orchestrates a complex multi-phased immune response. Together, target cells employ intracellular innate antiviral mechanisms to contain viral infection and replication. As immunologist of viral infectious diseases, an expertise of ours is to investigate immune correlates of protection from infection versus susceptibility and progression to disease. Over the last decade, we significantly contributed to the characterization of several immune-related pathways relevant in the antiviral immune response. In particular, we have been investigating the role of cytokines and chemokines production, pathogen recognition receptors, the inflammasome, the interferon-stimulated genes, and the regulation of the cholesterol metabolism as one of them, and the antigen presentation mechanism, among other pathways. Since the current SARS-CoV-2 pandemic first raged, the urgent need to define relevant immune correlates and dissect infection and immunological mechanisms has been mandatory. We committed to quickly scan a wide range of immunological pathways putatively involved in the antiviral response against SARS-CoV-2. We diversified our researches on different models. Lung biopsies as relevant tissues of a respiratory infection. Ex vivo peripheral blood mononuclear cells. And finally, we pursued the characterization of long-term immune response in a cohort of women who just gave birth. In this first project I would like to present you, we characterized the immunological features of lung removed from the first 2 Italian subjects who successfully underwent emergency bilateral lung transplantation due to COVID-related acute respiratory distress syndrome. Removed lungs underwent histopathological evaluation. Gene expression profiling for immune-related signature was performed on lung biopsies and SARS-CoV-2 stimulated peripheral blood mononuclear cells. Moreover, cytokine levels were measured on plasma and on bronchoalveolar lavage specimens. Microscopic examination of the removed lungs on the left showed widespread parenchymal thickening and signs of fibrosis. At the microscopic level on the right, a moderate inflammatory infiltrate was present in both cases, mostly in the form of perivascular T lymphocytes with a prevalence of the CD8 positive T cells compared to the CD4 positive T cells component. In regard to the B cells infiltrate, scattered CD20 positive lymphocytes were observed. On the left, lung from COVID-19 subject displayed an immune-activated profile. Such hyperactivation was evident in lungs from both subjects, with a sharp upregulation of genes involved in different aspects of the antiviral immune response. The signature included effector cytokines and chemokines, pathogen recognition receptors, immune activation receptor, inflammasome components. Moreover, interferon-stimulated genes, including cholesterol metabolism, were greatly overexpressed. This is a relevant piece of data, as there is a lot going on about interferon in COVID-19 patients who often are surprisingly lacking plasmatic interferon alpha protein in severe to critical cases. Furthermore, in the center, this hyperactivated profile was more evident in lungs from subject 1. Indeed, I would like to draw your attention on interleukin-6, IL-6, which greatly varies between subjects, together with the receptors involved in SARS-CoV-2 infections, such as ACE2 and the virus-interacting protein AGTR1. On the right, ACE2 expression in the lung parenchyma of COVID-19 lung transplanted subjects was evaluated at the protein level. Indeed, by immunohistochemistry, we found that ACE2 was present both in the alveolar space and the primary bronchus, besides being expressed by small vessels as expected. In line with the general expression pattern observed in lung tissues, plasmatic pro-inflammatory antiviral cytokines and chemokines were upregulated with higher level in subject 1 compared with subject 2. In particular, we found CCL3, interleukin-1, interleukin-6, IP10, and MCP1 being highly expressed among other targets. Unfortunately, we couldn't retrieve bronchoalveolar lavage from subject 2, and here is depicted in the center subject 1 compared to a non-COVID related lung transplantation. Analysis performed on the bile fluid showed that chemokines such as CCL3 and CCL5 and the pro-inflammatory molecules IL-1 and IL-6 were significantly higher in the bile from the COVID-related subject. Specifically, we observed a common signature in the plasma and in the bile of upregulated signaling molecules involved in macrophages activation, which included chemoattractant cytokines such as CCL3 and MCP1, and the interleukins and interferon. At the gene expression level, PBMCs from both COVID-19 lung transplanted subjects displayed a strong immune activation profile, both at basal condition on the left and upon stimulation with SARS-CoV-2 specific antigens on the right. Notably, such hyperactivation status was higher in subject 1, with upregulation of effector cytokines and chemokines, pathogen recognition receptors, immune activation receptors, and inflammasome components. Together with the gene expression data, the quantification of the cytokines in the supernatant from the culture of the PBMCs on the right confirmed that circulating lymphocytes from both subjects were characterized by a pro-inflammatory phenotype. In particular, high levels of monocytes macrophage chemotactic and activating factors such as CCL3, 4, and G-CSF, and the pro-inflammatory cytokines such as IL-6 and TNF. Briefly, in conclusion, we found that histopathological analysis show an extensive fibrotic remodeling accompanied by an immune infiltration. mRNA profiling on lungs and PBMCs show an hyperactivation on inflammatory pathways. Cytokines and chemokines profiling in plasma and in bile confirms the pro-inflammatory profile as seen by mRNA analysis. You can find the results and others in the published paper reported here. In the second study design, our aim was the profiling of the immune response triggered by different microbial and inflammatory stimuli. In order to do that, we used inactivated SARS-CoV-2, HIV, and flu viruses together with interferon alpha or IL-1 beta stimulation and exposure to CMV antigens or bacterial antigens. The cellular model employed was PBMCs isolated from healthy donor in this case. We used the Quantigene technology to profile the specific activation signatures related to each tested pathogen. Notably, the gene expression pattern was partially shared by all the stimuli employed and partially pathogen-specific as summarized in the reported heat map. In particular, following inactivated SARS-CoV-2 stimulation, a significantly higher transcription rate was observed for chemokines such as CCL2, 3, 5, and interleukin-8, cytokines such as IL-1 beta, interleukin-6, and interferon gamma, inflammasome components such as Qpcr and Cas1, and several interferon-stimulated genes such as MX1, EFIT1, and 3. This approach, therefore, allowed us to quickly point out the modification triggered by pathogen exposure at transcriptomic level in healthy control, even in absence of an actual infection. Owing the lack of time, I don't want to go deeper into details, which you can find here in the published manuscript about a broader study focused on antigen presentation-related mechanisms. As third and last study, we investigated the immune response against SARS-CoV-2 in pregnant women. It is well established that pregnancy induces deep changes in the immune system. This is part of the physiological adaptation of the female organism to the pregnancy and the immunological tolerance toward the fetus. Indeed, over the 3 trimesters, the suppressive T-regulatory lymphocytes are progressively more and more represented, while the expression of co-stimulatory molecules decreases over time. It is not uncommon that the less responsive immune system during pregnancy develops a suboptimal long-lived response. Moreover, such adaptation relates to an increased risk of infection and progression to severe disease in pregnant women. In fact, the suboptimal immune response is harder to be triggered, resulting in a lower ability to control the invading pathogen. However, upon a missed pathogen clearance, the pro-inflammatory stimuli build up, and this may then generate an intense, uncoordinated immune response resembling the inauspicious COVID-related cytokine storm. Of course, this graph is an oversimplification taken to extremes, and it should not be taken for reference. In order to perform this study, we enrolled 47 pregnant women who acquired SARS-CoV-2 infection during the late 3rd trimester, within 2 weeks before delivery for most of them. Upon delivery, blood was drawn at T0 and at the following time points at week 2, 4, and 24. Plasma was obtained to measure the anti-SARS-CoV-2 specific IgG and IgM antibodies to perform the actual neutralization titer and to perform plasmatic and cytokines and chemokines profiling. Moreover, PBNCs were isolated for mRNA profiling upon SARS-CoV-2 ex vivo stimulation. At the 6-month follow-up, in the top left panel, 80% of the subjects still were found to be positive to specific anti-SARS-CoV-2 IgG. Panel on the right shows the amount of the antibody produced peak between the 2nd and the 4th week, to then decrease over time. At the bottom, you can see that the neutralization titer nicely matched the amount of antibody produced. Indeed, this is not granted, as not everyone develops neutralizing antibodies, as you could also tell by the subject here in gray. Then we profile the mRNA expression of a number of immune and antiviral-related genes upon in vitro SARS-CoV-2 stimulation, in this case normalized upon over the non-stimulated ones. The immune activation characterizing the first time point resembled the one observed in the previous experimental model, but then it waned over time. Confirming the mRNA data, here the plasmatic level of cytokines and chemokines, marked with an asterisk all the proteins that significantly decreased over time. Overall, we observed the subject who got SARS-CoV-2 infection during pregnancy developed an efficient antibody-driven immune response comparable to the general population. mRNA profiling on PBNCs shows an hyperactivation of inflammatory pathways, which then fades away over time. Cytokines and chemokines profiling in plasma confirms the pro-inflammatory profiles, as seen by the mRNA analysis. These data are confirmed by flow cytometric assessment of the T-cell-driven specific response and immunological memory, which are not shown here, but which we hope you can find soon in the full published manuscript. Currently, we are planning to take advantage of the scientific information we quickly gathered, also thanks to the methodology we finely tuned, to unravel other interesting scientific questions, such as the immune profiling of other peculiar cohorts or the role of SARS-CoV-2 accessory protein on host gene expression, among other projects. I would like to thank University of Milan and our collaborators, such as Luigi Sacco Hospital in Milan, Policlinico Hospital in Milan, and Thermo Fisher Scientific. Thank you for your attention, and we will now be happy to take some of your questions.
And thank you, Premnath and Claudio, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some great questions coming in from our audience members. Our first question, concerning the pregnant women study, did the enrolled women display different levels of severity in COVID-19 disease progression? And if so, did you analyze the antibody production in the subjects stratified by disease severity? And was there a correlation?
Thank you for the questions. And indeed, we have different subjects representing basically the whole spectrum of disease severity. The majority of them was asymptomatic, but we had also some cases of mild to severe to critical disease outcome. And indeed, we performed such analysis, although owing the lack of time, we didn't show them in the presentation. And the results clearly showed that the amount of antibody produced did not match the grade of severity because of the widespread of the measured antibody titer. However, considering the percentage of subjects producing antibodies, we saw that while in the symptomatic group, 50% only of the subjects produced antibodies, into the mild to severe to critical group, basically 100% of them developed antibody-specific anti-SARS-CoV-2.
Thank you so much. And what is the sensitivity of the QG assay?
I'll take that. Thanks for the question. So for the quantigene, the quantigene exists in two different formats. The quantigene plex assays are so the sensitivity is about 1,000 RNA copies per sample per target. And if you go for a single plexing method, then it becomes 5 times more sensitive, so approximately 200 copies of your target RNA. So that is the amount of sensitivity for these assays.
Thank you very much. And can purified RNA be used in the quantigene assay, and how well do the results correlate to other gene expression measurement methods such as Qpcr, microarrays, and NGS?
Mm-hmm. Yeah, that's a good question. So yes, the biggest advantage of quantigene assays is that it enables the users to directly go with samples such as the whole blood, tissues, either fresh frozen or even FFPE embedded. But apart from that, if one has access to a purified RNA sample, of course, this can also be used to do the multiplexing measurements. As for the comparison with the standard gene expression methodologies such as Qpcr and even sequencing, there are studies and publications available which nicely shows that the quantigene assay data nicely correlates with the existing methodologies as well. So we very often get a very high correlation of more than 90-95%.
Thank you so much. Now, is there a data analysis package for the quantigene assay?
Yes, there is a free of charge data analysis tool that is available to be accessed by users on the Thermo Fisher cloud. If one has a basic Thermo Fisher account, then you can simply log in and have access to this tool where you directly upload your raw data in CSV format, and there is a place where you can set up your plate layout, name them, group them if you like, and choose the relevant housekeepers that are already built inside your panel, and do a relative quantification analysis.
Thank you so much. Our next question, and I want to thank again our audience for these great questions coming in. Our next one is there a protein multiplex assay as well?
That is a good question. Yes. So the Luminex platform offers not only RNA multiplexing, but also protein multiplexing assays. So we at Thermo Fisher have a long-standing experience of developing both RNA and protein multiplexing assays. And when it comes to protein assays, the largest available panel is about 65 plex, meaning 65 targets that can be measured in a single reaction. And very often we see users are happy to sort of match the protein profiling with the mRNA profiling. So starting with a protein assay and finishing with a quantigene, or vice versa. So the answer is yes, you can also do a protein multiplexing profiling using the Luminex technology.
Thank you so much. We have time for a few more questions. Our next audience member wants to know, cases of co-infections with different variants, are they known?
Claudio?
Yes, that's a good question. And among the study we performed, we did not focus on that. And in literature, some are reported, but I don't have any details right now to answer this question.
Thank you very much. And can you share a little bit about the panels available for other animal models like ferrets?
Yes, I can take that one. So the one other advantage of quantigene assays is that you can literally design panels for any species as long as there is a coded RNA, mRNA. We can design panels for those targets, regardless of the species. So yes.
Thank you so much. Now, why do you think there are differences in cytokine expression SARS-CoV-2 virus and flu viruses?
Thank you for the question. First of all, there are different viruses, and the immunological pathways that they stimulate might be different. But also, in the experiment we showed before, cells were PBMCs isolated from healthy donors. So for sure, they were already exposed to flu viruses in the past, and usually we use it as a positive control because almost everyone has been exposed during his life and therefore has a memory, immunological memory ready to be triggered. While as they were healthy donors, that was probably the first exposure of the cells to SARS-CoV-2. And so the immunological response is based on totally different pathways.
Thank you so much. Now, concerning the lung transplantation study, subject 1 displays a higher pro-inflammatory activated profile all along. However, in the last slide, the ex vivo stimulated PBMCs, the cytokines profile evaluated as protein released in the supernatant does not match what observed at the mRNA in the very same cells. How do you explain such apparent discrepancy, in particular regarding the IL-6?
So we noticed that, and as you noticed, subject 1 is more activated. His expression profile was higher basically in any condition at the protein level and at the mRNA level, both in lung and in PBMCs, even in basal condition with no stimulation. Probably the cells from this subject were already activated, and the in vitro stimulation, which is quite strong by the way, produced probably an hyperactivation resulting in exhaustion of the cells. And this phenomenon is quite common in severe cases of COVID-19, and this has been already observed and published, and we are also going deeper into this kind of analysis, and we often see exhaustion from the cells. And so they do produce a lot of RNA, they do respond to the stimuli, but due to an uncoordinated and overwhelming stimulation, they basically do not, it doesn't result in a productive expression at the protein level.
Thank you so much. Now, could you share what were the exact materials and methods for the immunological profiling of SARS-CoV-2?
So this is kind of a general question. I don't know if it refers to any particular study I presented before. Generally speaking, all the material methods are published in the paper I cited before. And we based our experiment on the Luminex technology that works either for mRNA expression with the Quantigen kit and for the protein expression with the multiplex for protein. So that's basically our approach. And in order to do that, we stored 100,000 cells more or less per condition. They were, suitably frozen as they were after the experiment. And then we followed the protocol by Thermo Fisher for the Quantigen, which is really easy and straightforward and require a minimal handling of the samples. You can find all the details in the published paper.
Thank you very much. And again, I want to thank our audience for these great questions coming in. For the shown experiments, you probably employed the same Q gene array as there are some similar targets. Did you diversify then the arrays according to the type of study? And the next part of the question, how do you customize them? Would you suggest the Qgene technology from a user perspective? Which are the pros and cons, and how practical and user-friendly is it? That's a large question.
Yeah, that's a lot of questions in one. Anyway, I try to go over all of them. So at the beginning, when we basically employed the Quantigene technology really early on, basically in March, and was new to us, and so we set up the methodology with the help of Thermo Fisher, with a remote explanation by them. And at the beginning, we designed a custom array based on our previous experiences on other viruses and based on the first publication that were coming out at the time. So we basically employed this wide scanning profiling Quantigene basically on all our projects. And since it was quite successful and we gathered quickly a lot of data, we then kept going with the Quantigene, and we customized even further, tailored on each single project. So for example, the one on accessory genes of SARS-CoV-2, or the one dedicated for the pediatric or the pregnant women. So step by step, we diversified our needs, and it's really easy to customize. Basically, the array is developed by Thermo Fisher technicians, and we just gave the list of the gene with the specification of the particular gene or of the particular region, but they then developed all the in silico testing of our probes. And let me go back to the question a second. And so from a user point of view, so it's the pros we found is that this technology requires really a minimal handling of the sample, meaning that we were able to start from different kinds of specimens, such as biopsies, whole blood, RNA already isolated and purified, PBNCs, cell lines. There are different buffers according to the needs, but there is no RNA isolation, there is no retranscription needed, so it's quite straightforward. And I think that main advantage is that you can perform in a single test many targets, up to 80 or something, and you have the full plate of 96 wells. So of course, in like less than 24 hours, from the beginning to the end, to the analysis of the data, for 80 targets for 96 wells, it's quite convenient. And even if the price is, I mean, not very low, but considering the amount of targets and the amount of cells process and the amount of time that is spared, we found that it's really convenient. And of course, doesn't work for a couple of targets or a couple of samples, but scaling up with many targets and many samples, we found that it's quite friendly, easy to use, and fast, extremely fast.
Thank you so much. Now, in regards to those targets, can I look at them simultaneously? How many can I look at simultaneously?
I'll take that. So as of now, you can look at 80 unique targets per reaction, per sample. That's the maximum size of a panel that can be built. But what is nice is we recently also developed a high throughput format of the assay. So classically, we did it using a 96 well plate format, but we also recently have a 384 well format to improve sample throughput. So if you have multiple samples to run, you can have them either in a 96 well or in a 384 well format. But the maximum number of targets is 80.
Thank you so much. Now, gentlemen, is QuantiGene semi-quantitative or quantitative?
So QuantiGene is a basically relative quantification method. So by default, it is relative quantification, but you can also make it fully quantitative if you have, for example, an in vitro transcribed RNA as a reference material that you can run as a standard curve and then quantitate your samples against it. So, but in a regular QuantiGene plex assay, it's by default a relative quantification method.
Thank you so much. We have time for one more question. If you stored plasma from COVID patients and use in stimulating normal PBMCs, what additional information might be gained if you also have stored PBMCs from the same patient, if you possibly know that?
That's an interesting question that we actually never thought about, and it is not part actually of our experimental setting, as we collect multiple samples and then we stimulate them in a controlled manner with a certain amount of virus, and it's going to be the same virus for each sample. So that's not our aim usually. And I would also say that since we measure mainly the antiviral response, it's not even given that, viremia for SARS-CoV-2 is quite rare, and it's only in severe to critical cases. So it's not our first choice usually for the aim we have. So it's not part of our experience. But that's an interesting point of view.
Thank you so much. Actually, we have time for one more. Considering SARS-CoV-2 is still so new, how long do you envision follow-up immune profiling studies like this taking place? It seems like one of the benefits of the QuantiGene assay you spoke about is a lice and go, and I can envision much more follow-up and additional organ characterization with all the long-term effects.
So I think that if I was able to reply to this question, I would have already the Nobel Prize. No, I'm just kidding. But of course, it's new, and we don't really know how long it's gonna last. We have characterized this cohort of women up to 6 months, and we are keep going. Actually, since 6 months was a few months ago, we are keep collecting data. I've seen in the literature that 1 year follow-up or 10-month follow-up is quite, studied already. And for sure, as the second part of the question is, yes, for sure, it's the QuantiGene is a suitable technology for the lice and go for additional organ characterization, and although it's not the experimental setting we usually use as we stimulate the cells, but that's probably really easy to perform with this kind of technology.
Thank you so much. And Premnath and Claudio, I want to thank you so much for your presentation today. Would you like to provide any closing remarks to our audience before we go? I'll start with you, Claudio.
Well, thank you. And I would say that it was a really interesting session of question and answer, and we covered many interesting topics. And we say that we, as said in my conclusion slide, that we are expanding the number of projects employing the QuantiGene, and this has been quite helpful all along, and we would like to thank also Thermo Fisher for their support, which was really useful for all of these projects I presented you, and for all the coming projects I hope you can soon find published.
Thank you so much, Claudio. And Premnath, would you like to add any remarks?
Yes. So firstly, thank you to all the audience for all their very nice and interesting questions, and also to you for hosting this session. So yeah, and a big thank you to Claudio, our collaborator, who has done some excellent work, and it was a pleasure working with him over the last several months in getting all these papers and interesting results out. So yeah, we are very excited that Quantigene was able to help Claudio and his team to come up with some very interesting observations in this study. Yeah, that's all I would like to say. Thank you.
Well, I want to thank you both again, Premnath and Claudio, for your time and for your important research. And I also want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for writing on today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions, and remind you that questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay safe and stay healthy. Bye-bye.